The techniques of genetic engineering used to produce factor VIII and other human proteins are highly complex, but the basic concept is simple. First, isolate the section of DNA that codes for the protein you want. Second, insert this DNA into living cells, usually bacteria. Third, grow the cells in a nutrient medium so that they express, that is, produce, the protein. Finally, separate out and purify the desired protein. Factor VIII has been produced by several different genetic engineering methods, and we'll look at just one of these. The first step of isolating the allele, of separating it out from all the other genes on the chromosomes, was done using techniques discovered in the early 70s. A crucial discovery was that certain enzymes cut the DNA molecule. They were called restriction enzymes because the effect of each one was restricted to just one particular sequence of bases on the DNA. Not only that, some of these enzymes cut one strand of the DNA several bases down from where they cut the other strand. This left the DNA fragment with a single-stranded tail. It's sometimes called a sticky end, because if it comes in contact with another tail carrying the complementary sequence of bases, the two pieces will stick to each other. Meanwhile, other scientists were studying small loops of DNA in bacteria called plasmids. They found that this DNA had basically the same double helix structure as the DNA of humans, animals and plants. It could be cut with restriction enzymes, leaving the plasmid loop open with sticky ends. Perhaps it would combine with DNA from other organisms. To find out, they treated the plasmids with the same restriction enzyme used to snip out a DNA fragment from a toad. When they mixed the toad DNA fragments with the bacterial plasmid DNA, the sticky ends joined to form new, continuous plasmid loops. What they had done was actually recombine genes, or create recombinant DNA. Even more startling, they found that when they inserted the recombinant plasmids into bacterial cells, the new plasmids would replicate normally, and the toad genes they were carrying were expressed. They produced the protein they normally did.